So it's really an honor to be here. This is my first society, my home society. So to be, while I dabble in a lot of other areas now, to be recognized by my very first home society is truly an honor. I also need to apologize. I put together a slideshow that's very different than what I usually do. Rather than go into any story in depth, I sort of concocted a lot of stories, way too many stories, trying to give you how I'm thinking about things, and I hope it will come across well. The second apology is I'm here with my daughter, and last night we went to Paul Bocuse, and for those of you who don't know what that restaurant is, you Google it, the experience of a nighttime, and I drank way too much wine, and then they gave us dessert cocktails, and let's see how this goes, but my apologies in advance. It was a wonderful night. So it all started actually with ISMB 2001, if we mentioned it, where this was actually the first time I gave a public keynote, and not keynote, just a public talk, and I was terrified, but there, we came across that molecular influences can really be statistical dependencies. And then during my postdoc in 2005, we realized that if we treat each individual cell as an example, then we can actually learn molecular networks. And the idea of the power of single cells and treating each individual cell as a sample, giving us enough samples to learn, for example, patient-specific disease networks, really changed my trajectory and what I was trying to do. Right now, of course, the HCA has tens of millions of cells, so there's much we can do. But at the time, in 2005, there was really no technology. So it was a search for technologies and a little bit waiting to really utilize the power of single cells until together with Gary Nolan, we introduced mass cytometry. This allowed us to look at about 30 to 40 proteins simultaneously in individual cells. And looking at that data, the strongest signal in the data is really a switch from discrete toggles of developmental lineages to really continue. So what we saw, the strongest signal in the data was a developmental continuum. In this case, B cells. This is the first principal component. Statistics 101, the dumbest method, and even that picked up that the strongest signal in the data is a continuum of development. Since then, there are probably 1,000 pseudotime algorithms, if not hundreds. But of course, the devil's in the details, as I'll try and show you, and pseudotime and trajectory inference is going to be a motif in this talk. But beyond just having a single trajectory or a single one-dimensional progression, looking at this data, we see that it actually has a beautiful geometry. In immunology, different subsets, if you know exactly what markers to gate them on, you can gate them on specific gates. If you try and use PCA, it all gets smushed together, but there's a nonlinear reduction that actually captures this geometry. And actually, single cell data and cell states reside in this sort of high dimensional geometry. Now, you don't want to overinterpret a two-dimensional map, ever. I call this T-SNE-T reading, and it can get you, just as good as some, T reading that some person does. So really a way to represent this is this phenotypic manifold is with a neighbor graph. This sort of reduces the dimensionality or makes you dimensionless, where each cell is a node in the graph and you connect the most single cell. This gives you a representation, a mesh for this phenotypic manifold, which you can then explore computationally. Community clustering on this gives you clusters in a very, and cell types in a very distinct way. And now, using this graph, you can search for trajectories as paths along this graph, really looking at local steps and similarities to find a global structure and global path through this graph. Now, once you have a trajectory, once you can order all cells along this trajectory, for example, a maturation or a differentiation trajectory, then you can do a lot of things. If you've measured, for example, single cell RNA-seq, you can actually order and the timing of all events. This is an example from, oops, why is this not working? Okay, this is a switch from Mac to PowerPoint, but you should have seen here just, a trajectory of all the rise and fall of all the transcription factors and chromatin modifiers that change during EMT, and I hope we won't have any more snafus in the move from Apple to Windows. So, as I said, the devil's in the details. If I first showed you a trajectory of B-cell development in PCA, that wasn't good enough. It took, since then, 3 years, and this is work by Elad to develop Wonderlust. And this allows us to get very, very accurate trajectories. This allowed us to order the cells with such accuracy that we could pick up a really rare population, 3 in 10,000 cells. And this is the population where the DNA gets shuffled and B-cell receptor is made. Once we organized this and looked at what's going on there, we could actually see that just in this tiny population, 3 out of 10,000 cells, there's a completely rewired checkpoint. Signaling is completely different in this trajectory. And this is the population that goes awry in pediatric leukemia. And we wouldn't have been able to get it if we didn't make efforts to capture rare cells. And this is going to be another motif in my talk, that a lot of machine learning methods, they go for the average. A lot of benchmarking goes for the average, what I call the boring populations, where nothing really great is going on. But actually, stem cells, rare metastatic cells that actually eventually kill the person, these are the most important cells. So when you develop computational biology, you can't take, machine learning off the shelf. You really have to think, am I capturing the rare cells? Many of the methods would treat these as outliers, and that's where all the biology is. Fast forward quite a few years forward. I'm going to skip, a lot of work with pseudotime. As a field, we were working a lot to try and find branch points. So going from a single trajectory, we wanted to go and see where cell fate choices were made. And there was a long time in the field where branch point was what all of us talked about. But one really needs to look at the data, and that was a little misguided event, because if you look at the data, it looked more like branch planes rather than branch points. Just like cell states were a continuous progression, a big realization came that cell fates should also be treated in a continuous manner. And this is where Palantir was born. This is work by Manu, who's now at Fred Hutch. And basically, again, now we want to treat cell fate and cell decision as a continuous process. And so we're going to orient our graph. We could use any pseudotime algorithm to take our undirected neighbor graph and orient the edges. And then by renormalization, we can create a Markov chain, and then take all the wonderful math that Markov chain has for us. And now if we model cell fate as a probability vector, we can say, what is the probability from any given state to reach each one of the terminal fates by counting all possible walks through the graphs. Now, counting all possible walks through a graph, if you're working in a sort of graph representation, can be pretty dicey, but that's where the duality between graphs and matrices comes in. So you can take neighbor graph, turn it into a cell-by-cell matrix, where each entry into the matrix is the weight of the entry between cell I and cell J. And now, once we've built this matrix, it's simple. You power up the matrix to go and look at paths of length 1, 5, 50, 500. You keep powering this matrix, and actually there's a closed form solution to reach each one of the absorbing terminal states. And now we can very quickly, with a very simple closed form solution and some matrix algebra, get the probability of going from each cell to each terminal fate as a probability vector, which is actually, what we call the plasticity or the potential of the cell. And you can see that cells have a lot of plasticity. There are very few cells who are really defined as they're developing. It takes time before they have biases before they finally commit. Now, here I'm actually going to go a little bit under the hood, because a lot of these ideas, they're very simple in concept, but you can get them all wrong if you don't really think about the ideas. Remember, I was telling you about rare cells, and these rare cells actually create huge non-uniformity in this phenotypic manifold. The hematopoietic stem cells, for example, are 5 orders of magnitude less frequent than the T cells. So again, if you use standard machine learning, sample cells randomly, you're going to get these, biased samplings in these very, very dense and boring regions of your phenotypic manifold, and you're going to miss all the transition areas, which are typically rare. You're going to actually miss anything that's important. So here we developed min-max sampling, and min-max sampling says, let's not pick the next random cell. Let's pick the next cell that is most different from any cell that we have, that captures regions of the phenotypic manifold that we haven't yet captured. And just to give you an idea of what this looks like, this is on endoderm data. This is on developing data of the early embryo in the mouse. And you can see, again, this differences in density. This is now colored by density. Red hot is very dense. Blue is barely any cells there. And a typical method would just go in this sort of red hot boring area, missing all the transitions, but min-max sampling covers the entire region and allows us to really interpolate and go over all the regions and all the transitions and all the paths that cell can take for their fates. Now, when you're working on biology, you really need to be thinking about your questions. I really let questions and biological questions guide my modeling assumptions, my algorithmic choices. And here, one of our big goals was to understand embryogenesis. How do we form? How do we start from a single cell, define our germ layers, and eventually, branch off? I love the endoderm, which creates the lung and the gut and all the fun stuff, the liver, the pancreas. And this was a collaboration with Kata Gentinakis. And this is what happens when you have a biologist and a computational biologist working hand in hand every day. So here, Palantir really rewrote the biological textbook. Here's a case where you have the earliest decision possible, the decision whether to be epiblast, to actually form the body, or to be extraembryonic and to have all these yolk sac and all these other tissues that support the embryo. Running Palantir and pseudotime and fate potential and fate probability, you can actually ask, okay, what happens as the cell choose to become extraembryonic? When do the cells choose to become extraembryonic? And because we could get both a continuous metric for progression, pseudotime, and a continuous metric for cell fate choice, we can see right here, as the cells are choosing to become extraembryonic, they are combinatorially expressing both FGFR1 and 2. So it's a combinatorial decision, and we could really use this approach to identify the regulators of this earliest decision, which were validated experimentally. If you go one day forward, we saw something really weird. We saw actually that a bridge between embryonic and extraembryonic, these cells had supposed to be split, decide, and never change their mind, but it seems like these cells were changing their mind. Looking at Palantir, there was again, at this point right here, which is actually now in day 5, after a full day and a half after they've already made the decision, there's plasticity again that suggests that these embryonic cells, epiblasts, decide to become extraembryonic somehow. Our colleagues, Kat and Sonia, thought we were crazy. We had to do a lot of statistical tests to convince ourselves that this is real signal in the data. And then using 2 lineage tracing approaches, we validated that this is actually true. This is something that no one would have ever guessed if Palantir and the data wouldn't have shown us this direction. And in this collaboration, and as I said, I'm not going to go into any paper or method in detail because most of it's published, we really rewrote the developmental biology textbook, finding 5, paradigm shifting ideas. Now, one doesn't rest on their laurels. I mean, I'm a very critical person. People in my labs knows I'm critical towards everyone and everything, and most critical towards myself and my own methods. And even though I just told you how awesome Palantir is, you have to think, okay, what are the limitations? That's how you move forward. And the problem is pseudotime algorithms make very strong assumptions. They make assumptions that, the cells only go forward in maturation, they don't go back, and that we know the starting point, which isn't always the case. It's often the case in development. But this is broken in regeneration, and this is also broken in cancer, 2 things I care about and study deeply. So the 2 areas, particularly cancer, that I study, the core assumption of these algorithms is broken, so we need to try and do something else. And Markov chains are actually very flexible. Many ways to build it. We can change the way features are built. And here with Prisca, we're actually, rather than looking at gene expression in single cells or protein expression in single cells, we're actually looking at imaging at entire organoids. And now we're doing a progression of organoids using imaging features. And this, it was able to get us a regulator of symmetry breaking. But more important for our case, we can change the way the edges are oriented. We can use other things other than just cell-cell similarity to say edges are going in one direction or another. This was a wonderful collaboration with Fabian Theis, who's a new fellow right here, and Marius, who's somewhere in this audience, I think, that came to visit the lab, and it was to marry RNA velocity and all the powerful things of the neighbor graphs and the Markov chain. And velocity too suffers from a lot of noise. So there's a lot of hype around velocity, but if you look at the data, often when you see these flow charts, there's averages of 500, 1,000 cells. When you look at the individual cells, you see they're actually very noisy, and sometimes the velocity are pointing outside the phenotypic manifold where there's no possible cell phenotype. So the idea here is to constrain velocity to the manifold and actually use velocity to orient our edges and orient directionality in our neighbor graph. So we compute the velocity and now weigh the directions, not only based on cell-cell similarity, but you're going to go to the neighbors, you're more likely to go to a neighbor that's pointing in a similar direction to the velocity. So we can create a new Markov chain, which uses velocity to infuse the graph with directionality. But then once we infuse the graph with the directionality, we have all the power of Markov chain, all the robustness of this global propagation, this global computation. We can actually say, okay, these regions, we have much more confident velocities. We're going to let them count more than uncertain regions. And we're able to compute global dynamics with the Markov chain infused with directionality of velocity. And here, again, things progress. There's lots of savvy math tricks to make this much faster, much more accurate than the original waypoint algorithms that we used in Palantir. Through the idea of metastable states, we find, we sort of cluster and break down the dynamics, which automatically finds initial states, so we didn't have to know them in advance, terminal states, and intermediate states using this clustering approach. Actually, ground truth in biology is really, really, really hard. It's very hard to find one. Here's an example with cell tagging, and as you can see with the flow charts, you barely see anything. But because we have cell tagging, we know future fates of some of these clones, and we can see that cell rank actually does a pretty good job if you're very close to the end, over 90-something%, but even 12 days before we get over 80%. But in vivo is much more complicated. So I'm going to now bring Cantor, because as I said, my motivation is to learn Cantor, and this is another paper by Cassandra, who's also somewhere in this audience. And again, what is our question? We want to know how this normal, happy, healthy pancreatic cell derails into malignancy. And we wanted to look at the earliest steps, and for this we needed mouse models that can capture these early states, because from human data, we already see full-blown, very malignant, very advanced PDAC, which is a very deadly disease, which is typically diagnosed late. So with the mice, we could look at these very, very early states and see what they look like. And actually, the actual full-blown cancers had much less variety than these very, very early states right here. And so zooming in on the early states, just when they got oncogenic KRAS, a single mutation, a single base pair change with a little bit of injury, 1-2 days after injury, you already see a lot of structure, a lot of differences, but what is the starting point? I mean, looking at this weird picture right here, it's hard to guess the starting point, and even the field can't help us, because the field had a huge disagreement and controversy, and many people sort of suggested very different starting points for PDAC. So running cell rank, it actually said, well, you know what? There's not one starting initial point. There are 4 initial points. And looking at what these 4 initial points are, these initial points actually mapped to the entire controversy. So this is a wonderful result where computation says, you know what, you bickering people, you're all right. all these are legitimate starting points. There's more than one place that can start. CellRank was excellent at capturing the starting point, but it wasn't as good at capturing the dynamics of what happens after each starting point. These starting points were very plastic. They had a lot of options, and we wanted to understand what was going on and how they create all this dynamics. The fun part of something failing, even a method I like, is it gives an opportunity and a motivation to develop another method. And here I won't say much except give a plug to Cassandra. This will be presented here on Wednesday. And the idea is, rather than using splice to unsplice ratio in RNA, we're actually going to use a TAC-seq, and we're going to use a regulatory network to direct edges. So if we can infer the regulatory network, and it doesn't have to be that accurate, we can say, okay, this rough and tough regulatory network says that future cell states go this way, and we get our velocity vectors from the regulatory network and get dynamics that way. And there's a lot of details that matter here, and Cassandra will tell you all of them on Wednesday. And, there are just many ways, and just posted this weekend on bioRxiv, together with Fabian, Philip, and Marius, are an entire new set of kernels and new ways to direct and orient the graph in different ways. It's also much more powerful and quick. And my favorite new kernel is actually to mix real-time, a time series of multiple time points, where you can connect the dots between time points, but also keep that pseudo time within time points, which is really much more powerful than just connecting the dots across time points without also integrating the variation within each time point. Now, I talked a lot about trajectories and cell trajectories, but I want to stop right here and take a strong pivot, because I think the single cell field, at least, is really, it got so focused on cells and labeling cells and calling cells labels and projecting cells into these dimensions where we don't even understand what they are and what's going on and what the genes are. But you can't forget that genes are what drives the biology. Genes are the functional units, and it's really always important to go back to the genes. And so we wanted to go back to the origins and understand how gene programs are organized in these cell states. So this is, again, a great collaboration between Russell, a statistics student in my lab, and Thomas, who's an immuno-oncologist postdoc in my lab that is a trained and certified immuno-oncologist. And the idea was to deconvolve gene expression into interpretable programs. So each cell is basically not a vector of genes, but a vector of modular gene programs that the cell is trying to execute. And each cell state is basically taking these Lego blocks of gene programs and putting them together to see that current cell state and what it's trying to do. Now, the way to do this is factor analysis. And there's been a lot of factor analysis around where you basically take a cell matrix and try and slice and dice it into gene factors. The problem with factor analysis is it's a fairly unconstrained problem, and there's just so many different ways to slice and dice this metric and get gene factors. And again, the best way to develop a new method is find out, well, what are the problems in the current methods? And some of the current methods can't distinguish between batch and biology, are strongly influenced by cell type that dominates the signal, leading to many factors, if they're not a cell type, that are very hard to interpret. So it's not a bad thing to cheat and to actually say, well, we know what we want, so let's teach the algorithm what we want. Let's infuse it with the good stuff. So we said, okay, we're really good at classifying cell types. So rather than have factor analysis redo the thing and actually inferiorly to the way that clustering works, let's tell the factor analysis cell types. And what's really good about that is each cell type has its own factors that say, okay, this is your baseline expression, this is the average expression of the cell type. Now factors have to explain away differences from this baseline. In addition to cell types, we can actually say, well, we know a little bit of biology. I mean, this group right here, many people in the audience really developed the gene ontology. So we can give gene programs and gene signatures and say, this is interesting biology. It's not perfect, it's dirty, it's built on these old 20-year-old bulk resources, but this is sort of gene sets that sort of capture biology. And we can even give cell type specific gene programs, like only T cells are supposed to express a TCR. But the key thing right here is not just to leave it as sets, but actually use these gene sets to build an input knowledge graph. This input knowledge graph allows us to make the algorithm much quicker, and also allows us to modify these gene sets and modify our sort of ground prior knowledge and adapt it to the data. But as you can see, you know, we're super powerful, even more quicker than some of these sort of deep learning methods. Most important thing is, okay, we have an approach, how are we going to score it? How are good factors? And scoring is a balance between our prior knowledge. We want edges to match the factors. We want, if we have a gene set, if we have an edge in the graph, we want the two genes to be in the same factor, but we also want to decompose the matrix in a way that will match the data. And the balance between that is what gives us our likelihood score. And in order to reach this balance, we're allowed to modify the graph. We're allowed to remove edges, we're allowed to add edges, of course with a penalty to get the proper likelihood function. And importantly, we don't even know. I mean, one of the fun things about single cell data is they're like entirely new factors to be discovered. And we can say, you know what? After we explained everything we can explain with our known factors, there's still a lot of residual. Now disconnect and find some new factors. And so really defining this likelihood and that balance between prior and match to the data and how much we're going to balance the two is what matters. So to summarize, for the entire data set, Spectra says which gene programs are active in this data set. We can feed it many more gene programs and it will say these are the ones that are active. For each gene program, it adapts the gene program and adds gene programs, removes gene programs, and adapts the gene programs for this specific biological context, which could be very different from where we gained the gene program. And for each cell, it tells us what are the genes programs that are active here. Thomas curated an amazing immuno-oncology set of high-quality curated gene sets, and that's available for anyone to use. And first, again, getting ground truth is really hard, but here, Jason Bustonero created this ground truth of known perturbations where we know exactly what's going to happen, a single perturbation, so we could see how well we're doing. And Spectra picked up the right cell type, goes way high exactly into perturbation, LPS to the moon, TCR, interferon gamma. But you know, these are very strong, very pinpointed perturbations, so it should be easy. But actually comparing it to the other leading methods, XPMAP and slalom, says that even something that seems easy is hard. This gave us confidence to go into a real immuno-oncology data set before and after treatment of PD-1 in breast cancer to see what's happening before and after treatment in responders. And as I said, and we were using these questions as sort of motivators when we were developing algorithm, I always like posing the biological questions up front and central. Reactive T cells, this is what matters. Immunotherapy is trying to activate them. If we can get reactive T cells, we've succeeded. The problem is that exhaustion, the bad thing, T cells are exhaustion, and reactivity have gene programs that are so similar that all previous methods couldn't really separate them because the genes that participate in both processes and because the gene programs are so highly correlated. Now, macrophages, they're known to be really important. They determine whether there are going to be enough T cells there or not. It's really an important frontier, but it's a lot less known. We don't really know what's going on with the macrophages. We just know it's important, so we want to understand macrophages and their roles. So to see how we did, there's a T cell data set, and because the authors of this data set measured the TCR, we could see which clones are expanding, so we know which patients are responding, which aren't. We know which clones are expanding and which aren't. And you could see that actually the tumor reactive and the exhaustion, if you just use the input gene signature, they're perfectly correlated. They don't distinguish between responders to non-responders. You can't get much out of them. But when you run Spectra, it deconvolves them. It finds two distinct gene programs. It completely changes the genes. That's our ability to change the genes and find these two distinct programs where tumor reactivity really matches the cells that are expanding in the data. It also is much higher in the responding patients. So the patients that are responding, both before and after therapy, are much higher. And again, looking at other leading methods, do not distinguish between responding and non-responding patients, do not deconvolve the exhausted and the non-exhausted. And even in a ground truth data set where we know exactly which cell has the expanding clone, has the target that the T cell expands, we could see it perfectly picks up these red cells in this sort of test. And again, more powerful than just picking up the responding cells, now we can see what genes are involved. Spectra dramatically changed from the sort of input set of 8 to 50 genes. And these genes have a lot of new genes that were recently published to be involved in tumor reactivity. And these things were actually published while we were developing and running the algorithm, so they were new. And this replicates in a number of additional data sets, these additional genes that we learned based on one data set. Switching to the macrophages, this is where previous approaches and clusters failed. The authors of this data set tried to find a cluster that matches responders. It didn't exactly match responders. There were many problems with this cluster. There was no single cluster that actually matched responders. And as you can see, there's really no difference between responders and non-responders. This is actually one of the reasons that we allow to learn absolutely new factors, because macrophages are quite unexplored. Our factors could pick up these differences and the continuums in the macrophage space. But in addition to really explaining transitions in macrophages, we really wanted to focus on this entirely new factor that wasn't similar to any input gene set and actually had that disconnect parameter that said, do your own thing. And this one actually perfectly matched responders and non-responders. It has to do, with invasion, but also cholesterol metabolism and this whole interesting set of genes. And it's actually validated in a number of validation data sets. To scale it up, we actually went and looked at lung cancer, even without immunotherapy, to see, do we see these same factors, these same immune subsets in a different subset? This is 1.3 million cells, over 300 patients, and we ran this without batch correction. Because once we actually have these factors, we have these gene programs, we say this is the gene program in this cell, you actually don't need batch correction. And Spectra actually said, okay, this is NUC-seq, all the other ones are cell-seq, picked up a factor for NUC-seq batch and separated it out, explained it away. And all the factors that we found previously in the breast cancer data under TCR, under immunotherapy, the tumor reactivity, the novel macrophage factor, a couple of new metabolic factors, they were all found, the same exact factors with incredible overlap in the genes and in the new genes added with this other data set. And remember, Spectra has added many, many new genes, so these are new factors. And now that we have all these, over 300 patients, we can actually correlate factors with clinical covariates. So this is wonderful, but one can ask, when do linear approaches fail? Factor analysis is linear. It means it can be fast, it's interpretable, but linear sometimes works, but sometimes you need to go nonlinear. I showed you at the very beginning, there was a lot of nonlinearity. And where linear fails, in my experience, is derailment in disease. So this is work with Elham Azizi, who used to be my postdoc, that started in my lab. Now she's at Columbia and with a bunch of people in her lab, and Vincent, who also moved on to Montreal. This is a project that took more than 5 years, and we really wanted to understand how normal cells derail and become leukemic. AML is a disease of derailment, and you can see here this NPM1 mutation. It's an AML mutation in about 50% of the AMLs, and you can see that you actually see this derailment, this group of cells that just become leukemic. Now again, standard approaches, and this is SCVI, the favorite integration method, but try any integration method under the sun. Somehow derailment from disease, the shared factors of disease, there's a lot of, like if you're looking at hematopoiesis, like AML doesn't reinvent the wheel, it just slightly alters it, but it's different enough. So all these methods that try and smush data together, in this case SCVI, cannot integrate disease and healthy. And our key insight was, we see that this data is nonlinear. Let's become even more nonlinear. Let's boost the nonlinearity, and specifically, let's allow dependencies between the factors themselves. In SCVI, the factors become independent. We are actually going to allow dependencies between the factors and add effectively one more layer into the neural network, make it deeper. And this deeper network, it actually factorizes the data into two possibilities, into two latent layers. And then the next layer, that used to be standard SCVI, is allowed dependencies between them. So that, a picture is worth 1,000 words. You can see in SCVI, the latent layers, the latent variables, they're independent, but actually once you make it deeper, once you allow dependencies, you can see these dependencies in our Decipher algorithm. And Decipher, and this is one case, but it does it for multiple AMLs with different mutations. We can see that one of the major components of this P space is maturation, is development, that's the x-axis, and the other is derailment, is disease. And we can see all these NPM1 mutations up here in the y-axis derailment. But because we have two layers, we can actually use the Z layers, the standard layers now, not in 2 dimensions, but in 30 dimensions to capture more gentle cell state dynamics, including progression to the different cell states. Now, Decipher can also give us denoised gene expression from the cell state, and the fact that we don't integrate it means that it gets them in a way that's a lot more reliable, so we can actually model denoised gene expression, and this allowed us to say which genes are conserved between AML and cancer, and there were AML and normal hematopoiesis, and there were many conserved, and which ones are derailed. We found many derailed factors, and I'm not going to go into all the details, but we validated them both in bulk and using ATAC-seq, really pinpointing especially a bunch of transcription factors and inflammatory factors that explain the disruption. Now, inflammation was a big component of the disruption. So now we're going to go back to Cassandra's work and these initiating states by cell rank and look at inflammation. And if we look at which are the most plastic genes, which are the initiating states, we can actually find that the most plastic cells, and I'm not going to go into exactly how we computed that, it's all published, actually have a lot of inflammation, have a lot of cell-cell interactions. Cell-cell interactions matter, so we said, okay, we have inflammation, cell-cell interactions with the immune system, how do we map that? Cell phone DB gave us a total fuzzball. Pretty much every possible relationship that could exist, exists. We used modularity of gene expression, the fact that you don't want to look at a single ligand receptor pair, but you want to look at a module, speaking with a module to give us robustness, really narrowed it down to a small percent of possible promising relations. And one of them was a feedback loop between the immune system and the cancer cells. Zooming in, this feedback loop was a feedback loop between very rare cells, but actually, downstream of this feedback loop, it impacted 70% of the tissue, both the cancer cells and the immune cells. So we have a feedback loop between immune and cancer cells, or pre-malignant cells, that powers up and downstream influences 70% of the tissue, starting from these very small and rare populations. And again, we disrupted this loop, and disrupting this loop not only halted progression into more tumorigenic states, but actually exactly the cell types that we predicted would be downstream of this loop, and exactly how much they were affected was validated with this CRISPR perturbation. Again, showing you the power of computational powers. This was great. We managed to do a lot out of single cells, disassociated, but what we really need is spatial data. And finally, and this is again something I've been waiting for 6-7 years, The spatial methods have matured enough. So now a large effort in the lab is really to focus on the spatial methods. You can see one example in a poster tomorrow by Doron. You could also look at it in bioRxiv. But really now, the era is to develop methods for spatial data. Everything from processing the data properly, what's really missing is interpreting. And this I'm gonna whiz through like in 1 or 2 minutes, just to give you one of our early algorithms. This is to understand the interactions between lymphatics. These are sort of like vessels that you know, deliver things in the crypt, and the stem cells that build the gut. The gut regenerates, recreates everything every few days, and how gut regeneration, normal gut regeneration happens and is controlled, and why we don't constantly have cancers is actually, with all that we understand in this process, poorly understood. This is a collaboration with Elaine Fuchs's lab and a power team of experimentalists, and Tini Chu, a computational postdoc in my lab. Basically, we developed Bayes Prism. The trick in Bayes Prism is that we not only look at cell type fractions, but we actually assign in a Bayesian way each gene to each cell type. So we have cell type specific count matrices and the cell type fractions in a completely Bayesian manner. Of course, there's lots of details, it's published. And again, this matters, the rare cells, the cells that interest us. Stem cells, super rare, only a few % of the data. Lymphatics, super rare, only a small % of the data. So if you look at all these published methods out there that benchmark well, like Tangram, they utterly fail on these rare and important cell types. And because we were thinking of capturing them, Bayes Prism does really well and really accurately on a ground truth to pick up where the lymphatics and the stem cells are. Now, again, it's still very noisy data. We use the fact that the crypt is stereotypical to really aggregate all these stereotypical crypts. This gives us a lot of power. We have hundreds of crypts in which we can learn very fine spatial resolution. And we can actually, for each gene and each cell type, see how it changes as it goes up the crypt. This pinpointed relin, and relin is the gene that interacts with the stem cells and basically holds back its differentiation and keeps it in the stem cell state. When you disrupt relin, the cells differentiate and the stem cells are depleted. And what's important to say about relin is this is a gene that was like almost nothing was published about it. PubMed is empty. This would have never gone into a panel that anyone would have designed in the field. Because we went global, because we went unbiased, we could get this really surprising genes with powerful computation. So what interests me these days? I love plasticity and how a cell has intrinsic plasticity and functions and interactions within the tissue context to, you know, make cell decisions and to build entire acting tissues, and how cancer hijacks them. And this is what we're studying now. Needs a lot of new methods, so everyone is invited to join our lab. And of course, I told you a lot of stories, and you know, I'm okay-ish. I'm getting an award, so I might not be entirely dumb, but my real luck is having over generations, and right now, the most amazing people in my lab. So Manu, Elham, Vincent were amazing postdocs. All of them have faculty positions. Cassandra, Russell, Thomas, Tini, all amazing. Also Doron, great guests. Marius was great to have in the lab. Philip is awesome. Elham and our team, and also a lot of amazing experimental collaborators, Gary Nolan, Katajan Tanakis, Scott Lowe, and Elaine Fuchs, to mention a few that I told you about, and thank you very much.
Who's got a question?
I'm scary.
Not a single hand raised. If it's going to be the first one in that back corner, I am not running all the way up there. All right, here we go.
Thanks. Dana, great talk. I really enjoyed it. So you mentioned about the rare cell population. How do you go about validation? This is very challenging in computational prediction many times, especially when dealing with rare population.
So I'm going to change your question. Before even validating it, you know, there is a real problem because this is noisy data. So you have real outliers. So first of all, how do you model so that you have a chance in hell to distinguish outliers from the rare populations? And this is, again, there's no, magic recipe that works all the time. That's why you actually have to be engaged in the biology. You have to understand the biology, and you have to give the algorithm a little feature of the biology. For example, in one case, you want to look at rare cell types. In other cases, you enrich experimentally for the rare cell type when you're learning. In other cases, it's important sampling where you give more priority to your learning in rare cell types. And then how do you validate? Sometimes you know a little bit. In the case of the last example of that I showed with the IHC, this is where what we have is we took the visiums and we actually used immunofluorescence on the same visiums for markers. So we had immunofluorescence that marked, here are the stem cells. We had immunofluorescence with very distinct markers that marked, here are the lymphatics. So with immunofluorescence and clear markers, we knew where they were as a ground truth. Of course, the visium data didn't use that, but then we could look at the deconvolved visium and say, did each of the algorithms find stem cells and lymphatics where we see in the spot that they exist based on immunofluorescence? Many times, we just have, the most important thing is to learn new biology. So we say, we are confident in ourselves, we're confident in the signal, we have a decent track record, now go a half year, 8 months and validate it. And, for example, in the first case of these rare cells that actually, instead of continuing to be embryonic, become extra-embryonics. Kat and Sonia it took a bit of convincing to convince them to do that experiment, but that was, the sort of put your money where your mouth is and validated biologically, but that could often be, 6 to 12 months of labor. So you should be very confident and first see if there's any artifact or statistics that is misguiding you.
Hi, Dana. Thank you very much for this talk. I just wanted to say that I'm here.
Where are you?
Here.
Okay, yeah.
I'm no longer young, but you were still an inspirational role model for me. So thank you very much. My question is more to do with, I work in cancer, and of course we deal with non-responders to drugs, to therapies. So I was wondering what your thoughts are regarding using these methods or what we've learned from these methods to understand the perturbation of the drugs, especially the combination of drugs. Whether you think that we can learn how these drugs change the landscape, the polarity of the cells, the organization, and we can infer or maybe predict some sorts of direction of response.
So one has to be really careful in responders and non-responders because there's just a lot of variation. Cancer is an individual thing, so you have each individual cancer is very, very different. You have a lot of clinical covariates. In some cases it matters what hour of the day the drug was applied and what the person ate for breakfast. So the first thing you need, if you really want to understand the perturbation of the drug versus the not drug, and again, computation can only do so much. This is where good experimental design, a good experimental partner comes in. And I think the first thing we need, and we don't have enough of this, is a good clinical cohort that tries to match the responders and non-responders for everything that we can. And depending on how heterogeneous the cancer is, how many samples we actually need. In another project, which I didn't tell you about because we had very homogeneous cancers, we took a good matched cohort of 5 responders and 5 non-responders and really could pick up mechanism. But in other cancers, you really need tens of patients on either side to tease apart, the actual response. But I do think that single cell data with careful analysis and good clinical cohorts and enough statistical power, the field is wide open. Right now, most of the data out there is badly designed and small cohorts, and we need to overcome that, get bigger cohorts that are better designed, and then the discoveries will come.
Yeah. Hi. I have one more. Yeah. So, it was all fascinating, and there could be a lot to discuss. I have one question that was related to this PDAC controversy resolution. So you, if I understood correctly, right, you showed that the KRAS mutation basically introduces this PDAC state in those, from all those 4 cell types.
It's not exactly PDAC, it's pre-malignant, but yeah.
Pre-malignant PDAC. And do they use the same pathway, or does it hijack different pathway in each different cell type? Did you go in this kind of detail?
We haven't gone in this type of detail because I think we'd need more data, but the critical thing is not the pathway, and I think the pathway is fairly similar, but the ability to enter a plastic state. And one of the things that we see in a plastic state is, for example, we showed that plasticity is the ability of a cell to actually acquire many different fates. And what we saw, and again, I tried to give a narrative and didn't go into all the details in any one thing, and you're welcome to read the paper. But in the plastic state, what happens is actually there is an opening of chromatin at a lot of communication genes, a lot of receptor and ligand and cell-cell communication genes, their chromatin opens. So I do think that mechanism is shared of making these cells be more responsive and able to both influence and be influenced by the environment, gain plasticity. I think that is a very shared mechanism. What exactly happens beyond that, that's less clear.
We have a question that's been upvoted in the platform from Naoki Osato. I've heard about the zero inflation problem in single cell analysis. How do you think about it, and how do you deal with this problem?
So again, I don't like the term zero inflation, and a lot of the zero inflation statistics are very, very old, from older technologies, from Fluidigm, that's no longer used. So thinking about it as zero inflation and using the zero inflation statistics is wrong, and it's sort of gotten stuck into the field because that's what existed in the earliest thing. But what we do have is basically, the way I think about it is, depending on the technology and how deeply you sequence, you have all the transcript in the cells, and you are sampling 5 to 20% of the transcripts per cell. So methods that actually say, for each individual cell, I'm just closing my eyes and sampling 10% of the transcripts here, that's the statistical way to look at it. The real power is that you don't collect one cell, you collect thousands and tens of thousands and sometimes hundreds of thousands of cells. And if you find many similar cells, then you can say, well, each individual cell, I can't exactly see what's going on. And I have another method which I didn't go into, C cells, which actually aggregates into very fine groups these cell states that I think are the same, to give a pretty good bulk estimate of what that cell state is. But more generally, beyond C cells as one solution, is you've sampled only part of the transcripts, but you've sampled the landscape with many thousands of cells, and just do the statistics depending on the question that you want. There's always trade-offs, so you always have to think of what is your goal in your analysis.
Thank you. We have time for one final question.
Hello. So this is really cool work. I just wanted to know, in terms, as you mentioned, the kind of the functional unit is the gene, right? And when we analyze cell gene matrices with different methods, we don't really assign very high dimensional embeddings or pre-trained embeddings to genes. Have you thought of integrating methods like pre-trained embeddings for like the proteins that come out of genes or genes themselves to better inform your algorithms, or are we still kind of get stuck on just representing genes as a label?
Well, again, there's a big difference between proteins and genes, and our measurements for proteins are even more limited than the measurements for genes. So I certainly wouldn't go, the pre-trained protein models are very specific on protein structure and very specific protein-protein interactions and very specific context. So I don't think that they will help us. I do think that one of the big problems with all these embeddings that have become very, very popular is that they do lose a lot of the genes. They do get a lot of the genes wrong. They do often wipe out a lot of interesting biology because they don't distinguish between batch and biology. And, I think we're in a first generation of methods. And, for example, if we look at spectra, which is a very linear method, it at least said, okay, let's take the cell and re-represent it as a vector of, cell programs. So my embedding here was cell programs. It was entirely linear with all the linear limitations, but I did get a very robust low-dimensional embedding that way with cell programs. If we can infuse more cell programs in the right way, I think, that's gene programs in the right way, that's a way forward. XPMAP, makes the first attempt at it, and we're still in early days. So I think the field will improve with these embeddings, and people who are developing these embeddings should not forget the genes. A lot of the methods are currently being benchmarked at, I got the cell type right, that's fine, success. And the first thing is, our metrics, our benchmarking, and our training, and we need to go away from core cell type labeling, and everything else will follow.